Okay, so in this short video, we're going to look at population genetics for A2. So in terms of population genetics, as that suggests, it's looking at genetics at a population level. So in other words, we're looking at the organisms of the same species. Okay, so some terms you need to know. The gene pool is the sum total of all the genes in that particular population or of that particular species. The allele frequency is the proportion of a particular allele at a particular time, and the genome refers to all of the DNA of an individual, so of one species. Okay, the first part of this really is looking at the Hardy-Weinberg equation, which is used to, as it says, predict how gene frequencies are passed on or transmitted from generation to generation. It's based on a cross between two heterozygotes, so a big A and a small a, as you can see here, which they refer to as P or as Q. In terms of P is the frequency of the dominant allele, and Q is the frequency of the recessive allele. Okay, so if you get your big A, that's the same as the frequency of a small p and a small p, or p squared. If you get a big A with a small a, as you do here and here, that's the same as a PQ, so you have 2 PQ overall. And then if you get the homozygous recessive small a, that's a Q times Q, or Q squared. Okay, and then what you need to remember is P squared plus Q squared, sorry, P squared plus 2PQ plus Q squared equals 1, and also P plus Q are equal to 1 as well. So when you're trying out these questions, it's trying to interpret, are they asking about the frequency of the allele or the frequency of the gene, or are they talking about a condition or a phenotype? Okay, if they're talking about the phenotype, then they're talking about Q squared, as it does in this question. So here you've got 1 in 2,500 babies is born with cystic fibrosis. Okay, so that's leading on from, and it tells you that's a recessive disorder. So that would be, for example, small c. So that's giving you Q squared. Okay, so therefore Q squared is 1 over 2,500, which equals 0.0004. And then they're asking for the percentage of people in Northern Europe who are heterozygous. So the heterozygous is the 2PQ. Okay, so the carriers for cystic fibrosis. So if we've worked out Q as 0.02, the square root of Q squared, that means that P is 1 take away Q, which is 0.98. Okay, then you multiply 2PQ because you've got 2 of those individuals in that cross. So 2 times 0.02 times 0.98 equals that, which equals 0.04, which equals 4%. Okay, so that's an example where they're asking for the percentage of people. Other examples sometimes ask for the number of individuals in a particular population. So in this one, rhesus factor, so linked to blood groupings, individuals can either be rhesus positive or rhesus negative. The allele for rhesus positive, which is a big D, is dominant, so rhesus negative is recessive. So in this population, there's 400 individuals. It was found that the frequency of the negative allele, small d, was 0.15. Okay, so because they're talking about an allele there, or a gene it could be, they're talking about Q. So Q is equal to 0.15. Again, they're asking for the heterozygous or heterozygotes, but this time, as I say, they're talking about the number of individuals out of the 400. Okay, so Q is 0.15, P therefore equals 0.85, so 1 take away Q. So we're looking for the heterozygous ones, which are 2PQ. So 2 times 0.15 times 0.85 equals 0.255. Because they're looking for the number of individuals, then we'd have to multiply that by the total size of the population, which is times 400, which gives us 102. Okay. Another example here, you have Drosophila, which are little flies it was found that 176 of these Drosophila had vestigial wings, which you can see there's a part C previously in the question. They'd have told you that the vestigial wings was recessive, and 924 had normal wings. Again, they're asking for the number of Drosophila, so the number of individuals who are heterozygous. Okay, so the vestigial wings, there's 176 there, the recessive ones. So that's talking about a phenotype. So that's talking about Q squared. Okay, now we'd have to find the total size of the population. So you'd have your 176 vestigial wings, you have 924 normal wings, which includes the dominant and the heterozygous ones. That gives you a total of 1,100. So for Q squared, it's 176 over the total 1,100, which equals 0.16. Therefore, the square root of that gives you Q, which is 0.4. P plus Q equals 1, so therefore P equals 0.6. The heterozygous, again, it's looking for 2PQ. So 2 times 0.6 times 0.4 is 0.48, okay, or 48%. And then we'd have to take, because they're looking for the number of Drosophila, it's 0.48 times the total, 1,100, which would be 528. Okay. They often ask about the conditions that are required for Hardy-Weinberg equation, and those conditions are, you can see here. They often ask for 1 or 2 of these. So population is large, mating is at random, individuals are diploid, there's no selection, differential selection taking place, mutations not taking place, and no migration also. Okay, so it's just mentioning a few of those if they ask about the conditions that are required. Okay, the next bit of this topic is looking at sources of variation. Okay, and with this, we sort of revisit meiosis a little bit. So if you remember meiosis, in this case, we've got 2 chromosomes here. You'd normally, with humans, obviously be talking about 46 chromosomes, but 2 in this one. So during the different phases, your growth phase, your synthesis phase, the DNA replicates, so they form that sort of X chromosomes, if you like. Then you have prophase, where they line up in homologous pairs, okay? And that's where you may get some crossing over, so swapping of genes between homologous chromosomes, which may occur. Also, during metaphase 1, then they line up across the equator. And if you had your 23 pairs here, they'd be independently assorted or random alignment. Then those chromosomes at reduction division, chromosomes are pulled apart, cell splits, and then you have a second division which pulls the sister chromatids apart, which we can see here. And then you end up with 4 gametes, so 4 cells, which are different from the original cell in that they're haploid, so they have half the amount of DNA, and also you can see they're different from each other. So that can lead to variation. Okay, so as I mentioned there, independent assortment during prophase 1 or anaphase 1 of meiosis. You can just see if the blues were female, would have come from the mother, the reds would have come from the father. There's different permutations in terms of how these would line up. Okay, and if you think of 46 chromosomes, there's many permutations in terms of how each pair of homologous chromosomes will line up, and then when they separate, that will lead to variation, as you can see there. Okay, and the other one crossing over, you can see that you get that formation of the chiasma or chiasmata, gene swap positions. Therefore, you're left with different variants or different variations of that homologous pair of chromosomes. Okay, it can also be seen in cross-fertilization or random fertilization, in that it depends, these sperm, as we talked about before, because of meiosis, these will have different sets of genes within them. Depending which sperm fertilizes the egg, then that will determine which genes are put together. And if it was a different sperm fertilized the egg, then you would be different. Okay. The other types of variation then are brought about by meiosis, or sorry, mutations. And If we revisit protein synthesis for that, you can see here a section of DNA. From transcription, you produce messenger RNA, and then translation, it produces or translates that messenger RNA into proteins or polypeptides. Okay, and you remember this little diagram. So you have your ribosomes, you have your transfer RNA molecules, and that gives you the long chain of polypeptides or amino acids in particular orders or particular sequence, depending on the sequence of bases on the messenger RNA. Okay. Mutations are to do with changes in the DNA. So if there's a random change in the DNA, it's going to therefore affect the messenger RNA, which therefore affects the primary structure protein or polypeptide. Okay, so which amino acids are coded for. So for example here, this is a substitution. So there's our original base sequence that you can see. This one, GCG, codes for amino acid 3. If it was substituted, so the C was substituted for a G during replication, if something went wrong and it's substituted for a different base, then the amino acid would be different. And you can see here that codes for the same amino acid as the one down at the bottom. Okay. Some mutations are silent, in which they have no effect. Okay. Some or have a bit more of an effect, as you can see down below here. So if that's no mutation, if our DNA was TTC, our messenger RNA would be AAG, which would code for the amino acid lysine. If it was a silent mutation, it doesn't have any effect, so it could be TTT, so that C was replaced by a T. So you get AAA at the messenger RNA level. That codes for the same amino acid. That would have no effect on the protein. A sort of nonsense mutation, as they call them. If it's the first base that changes from a T to an A, that will give you a stop codon, which means that then the polypeptide would be a different length and could cause drastic effects, let's say. Or it could be something called a missense mutation, which we'll look at in a bit more detail in a second. So it gives you, this one's looking at the second base changing from a T to a C or a G. Therefore, the messenger RNA changes, and therefore the base that it codes for changes as well. Okay, and that can have effects, obviously, at a polypeptide level. Okay, the other time mutation is a frame shift mutation. So if there's deletion, so here we can see if this T up above was deleted during cell replication, then the amino acid TAC would be the same down below. ATA would then suddenly change to AAG, which would be a totally different amino acid. GCG, the G's moved to there, so that would change as well as CGT, different amino acid. TTA, the T's over here, so TAG, again, would give you a different amino acid. So every amino acid which comes after that deletion is different than what would be coded for in the original piece of DNA. So that's this part of the DNA, if you like, has shifted up the way to be part of a different triplet code. So that's what's called a frame shift mutation. Many other different types of mutation. You can have things changing around, you can have duplication, you can have different bases inserted. There's your deletion one or frame shift one. You can have translocation with bits of DNA moving. The main ones we look at are substitution and deletion. Okay, and there's your, and again, looking at your silent mutations, so ones that don't have any effect. All of these base sequences on your messenger RNA would all code for the amino acid leucine. Okay. We also have chromosome mutations, and as that suggests, it's a whole chromosome causing a mutation. So Down syndrome is an example of that. Down syndrome is an extra chromosome on pair 21. So we have 3 chromosomes here, which gives that person 47 chromosomes rather than 46. So that's an example of a chromosome mutation. Mutations can be caused by many different things: radiation, different chemicals, different viruses, can cause mutations as well. Okay, the next part of this then is looking at selection. So hopefully you'll recognize Charles Darwin, who was a sort of grandfather of the theory of selection or theory of natural selection or evolution. There's one of his quotes, which is worth reading. So we have to be adaptable to change, often referred to, natural selection often referred to as survival of the fittest. Now that doesn't mean you can run for the longest or lift the heaviest weight. It means that you are better adapted for that particular habitat in which you live. Okay, so the example here, with any population, you have variation within their genes, and that will cause, in this case, different colors of mice. Okay, some of these are reasonably fit or better adapted for the environment in terms of the darker ones. Some are less fit. Okay, so have poor genes to allow them to survive in the habitat. So those are selected against, in this case, by a predator. So the lighter ones get eaten. Then you have more of these dark ones around. They have the better genes that they pass on to the next generation. So you end up with a larger number of, in this case, the dark colored mice. Okay, two different types of selection that you need to know about. Most populations will have this sort of normal curve, okay, or sigmoidal curve. So there are a few people, organisms, whatever it is, plants at the extremes, whereas most of them tend to be average. Now this is fur color, it could be height, it could be anything else. Most people or organisms are found at the average. Okay, with stabilizing, something called stabilizing selection, you get pressure at both of the extremes. Okay, so it may be predators, it may be lack of food, lack of space, whatever, tends to be predation. There are pressures that cause the variation within the population to narrow, as you can see here, or there's your normal curve. We get selection pressures at both extremes, then your curve becomes narrower. Okay, so more of them are found around the middle. Directional selection is the other one you need to know about. That's where there's selection pressures at one of the sides or one of the extremes, and it causes generally your graph to shift. Could be to the left, in this case to the right, so the darker color of mice, similar to that last example. Okay, so there's your stabilizing selection, pressure at the extremes, and your variation becomes narrower, as you can see here. Also, that's stabilizing selection. Directional selection, where one of the extremes is selected against, so therefore the graph shifts generally to the right, could be to the left as well, depending. So that could be a seasonal change causes that directional selection, or it could be habitat change, for example, causes that as well. There's a couple of examples. So with robins that lay 4 eggs, okay, the mass of the eggs, you can see here is some will have very small mass, some will be larger mass. The smaller mass of eggs don't survive, they're not viable. The larger mass of eggs, it's harder for the robins to feed, so they're selected against as well. Most of the ones that survive will be of average mass. Okay, and the peppered moths is an example of directional selection, in that the lighter color was selected against due to the Industrial Revolution. So more of the moths found after that were found to be a darker color. Okay, and you can see that type of thing there. There's your light and your dark colored moths. Okay, also a little bit that they have in this is something called polymorphism, and we've come across that before looking at blood groups and our patterns of inheritance from genetic crosses. That's when you have more than 2 alleles which will code for a particular genotype or phenotype. So an example of that is your blood groups, where you have IO, IB, and IA. IA and IB are codominant. IO is the recessive one. Okay, and another example of many genes deciding the patterns can be seen in snails, the banding in snails. Another example of that is in sickle cell anemia. Okay, so sickle cell anemia is an example of a point mutation or one base mutation. You can see the normal red blood cell basis or base sequence is GAG, whereas that changes to GTC, which codes for a different amino acid, which then causes that person to have sickle cell anemia, which means the red blood cells don't carry as much oxygen. Where that is selected for is in places where you have malaria. Okay, so actually the sickle cell trait, where you carry the sickle cell gene, not where you suffer from sickle cell anemia, but you carry the gene, actually leads to 100% survival rates from malaria. Okay, whereas normal without the sickle cell gene only leads to 90% survival rates with malaria. Okay, so in places where you have lots of malaria, that sickle cell trait where you carry the defective gene is actually selected for. Okay, then we move on to look at what a species is. So a species, as you can see here, is a group of individuals with common ancestry, so similar features, etc. They are able to interbreed and produce fertile offspring. Okay, and each species is reproductively isolated from another species. So in other words, they are unable to breed. Okay, some examples of different species, you can see here a tiger and a lion. Those are different species. Now they can interbreed to produce something called a liger, but a liger is infertile. So these are considered a different species. Same with a horse and a donkey. They can reproduce to produce a mule, but again, it's infertile. So the donkey and the horse are referred to as different species. This leads on to something called speciation, which is the formation of a new species. Okay, and again, this requires reproductive isolation. Some of those isolations may be due to barriers to the actual reproductive process. So it could be mechanical barriers to mating or fertilization. So for example, two different species of dog, a Great Dane and a Pekingese, they have barriers to reproduction, as the pictures suggest. It could be that their offspring are infertile, as with the previous examples of the tiger and the lion there. And the reproductive cycles may not be in sync, so they can't reproduce that way. It could be due to behavioral isolation, okay, and that can be where there's different courtship rituals that it talks about here, and that's often seen in birds of paradise, where the male often has to perform for its mate. If it doesn't live up to scratch, then the mate clears off, which obviously means then that doesn't lead to reproduction. Okay, and also it could be ecological isolation in terms of they might be in different habitats or have different food preferences, etc. Leaf mining insects are examples of that. The example that we can use where you have geographical or allopatric speciation is Darwin's finches, so on the Galapagos Islands. So when he traveled around in his boat, he discovered that the finches on different islands had different types of beaks, and that was really due to their different food sources. So they had changed over a period of time, or evolved over a period of time to have different beaks to feed on the particular food sources that they had on those islands. Another example is the Grand Canyon in Arizona. You have squirrels that many years ago would have been the same species, but because they've been separated by this Grand Canyon and they've evolved in certain ways, they no longer reproduce to produce fertile offspring. So that's an example of allopatric speciation, where there's been geographical isolation, which has led to speciation, or different species being produced. Okay, hopefully that helped with population genetics.